This is not good. Make it full screen, it is not going further. So when we say sleep deprivation, what comes to your mind? Is it the absence of sleep, or is it something more? Okay, some people are saying it is more. So yes, so sleep deprivation does not always mean the absence of sleep. It's much more than that. So now there are many questions, especially in the current society. Is all the areas of the brain equally affected or equally involved, right? So we have just seen in the previous seminar that the homeostatic drive, it tried to recover the sleep in the shortest duration, right? But still we need to know several questions related to sleep deprivation. What happens in the disease condition? What happens in the neonates? What happens in the children? So there are many more questions for which we are looking forward to the answers. We don't have the answers for all, but we will try to share the knowledge which is available. So for that, we need to understand what is the function of sleep, because we will have several underlying little answers. So we know that it is also the final answer to the functions of the sleep or the role of sleep is still an open question. Any of you can have the research on it and can go for the Nobel Prize. The question is still open. So the homeostatic restoration, it is a role to play in the thermoregulation. We have the tissue repair. We have the immune system. When we sleep, it is getting improved. Then we have the memory processes. And of course, when we are sleep deprived, we have a lack of concentration, attention, performance. So this means this have a role to play in the sleep. So in order to know the role of the sleep, we have to go for the sleep deprivation methods. So in this diagram, what you see that sleep is not constant throughout the life. What you see here in this picture, a newborn undergo cycles of the sleep and the wakefulness. The upper portion, this is showing the time spent in the wake. So for shorter duration, they wake up, again go into sleep, wake up, and this cycle goes for 24 hours. So this is more or less a polycyclic sleep or polyphasic sleep, which you find in the children or the neonates. So the neonates are sleeping almost 85% of their time. Fine. And that sleep pattern, it changes when they grow. So by 1 or 2 years old, so it is still triphasic. However, in the night sleep is increasing. The duration is increasing. So further, 4 or 5 years old child, they will have the biphasic sleep. And by the time they are 10 years or they are in the teens, the sleep duration is longer. However, it is similar to what you find the monophasic sleep in the adults. And this you find in the adult. So sleep is a dynamic process, so which is age dependent. So whether the sleep deprivation during the different age group will have the different outcomes. So these questions which should be in our mind. This I have shown in the morning slide, so I can skip. In this picture, what is shown is that for the slow wave sleep, so this is the EEG during the slow wave sleep, you can see the synchronized waves, which is nothing but high amplitude and low frequency. And during the REM sleep and the wake, you have desynchronized waves. So that's a high frequency and the low amplitude waves. And this is the recordings of the cell. This is intracellular recordings. And this is EOG and this is EMG. So if we amplify a part of it, what you can see here, that the recording of the cell or the neuron, you see there is a hyperpolarization. So that is the one in the thalamocortical network. So that is the basis of getting the slow wave sleep in the during the non-REM sleep. Now when we talk of the sleep, we look into the functionality. For example, when we have the sleep-wake cycle, if you look at the biological function, coupling to the behavior state. So for example, this is the waking state or the wake predominant state, and the right side is the sleep predominant state. So the wake energy, so in this, when we are into the wake state, there is upregulation, because we have to have lots of energy to be utilized performing various functions. Fine. However, during the sleep, okay, so during the sleep, there is a down regulation of this particular state, which is the wake energy state. Because however, if we are into this, the predominant state, so there is called the biological investment, because that's the time when tissue repairing is happen. Okay, immune responses are there, and then we have, your parasympathetic drive is high, and we have synaptic plasticity that we have seen in the previous slide. So that is also down regulated. So many functions, so both the states are performing different functions. So in this picture, we are able to see the energy allocation in these two different states. And also if we see between the non-REM and the REM sleep cycling, so in the brain, if you look at the top panel, so this is the portion which is showing that our circadian rhythm are controlled by suprachiasmatic nuclei, and that is talking to the wake centers and the various sleep centers. So during the wakefulness, we have to have energy allocated for the waking effects, because our vigilance, the animals, for them that's a predatory component also, they have to avoid the predators, and also for reproduction they have to have the energy allocated, and also foraging they have to get their food also, they have to search for the food. So this requires the energy, lots of energy. So that is a thermoregulatory efforts involved in staying awake. However, if you look into the sleep, so that biological investment, because we see there is a growth, as was shown in the previous slides, that there was an increase in the growth hormone, and then we have the maintenance, we have the repair, and we have the neural networks reorganization in the brain. Now, what we want to see is that if we look at the various conditions, for example, if we go into the torpor, that is we get into a state of inactivity for very long, some of the organisms do that, or we stay continuously into the wakefulness, or if we have the sleep and the wake cycle, in which condition we will have the better organization of our energy management. So this one of the theory, which is the control of the energy allocation, which is shown in this picture. So for example, during the waking state, we have the hormonal control through the ACTH, which is very different from the sleeping state, where there is a predominance of the growth hormone, prolactin, and the LH. And also we have the local clocks, feedbacks, and also the autonomic responses for the various organs. And these are the ones, these different functions during the wake and the night, these cycling essentially help in the control of the energy management or the energy allocation. So these are the different perspectives of the looking into the functions. Now, for example, if we have the short-term sleep deprivation, let us say we have the one-day sleep deprivation, okay? We have the increased homeostatic drive. But if we continuously go into the sleep deprivation, let us say 3 days and the 4 days, our drive will slightly reduce. However, our deficits in the attention will be there in all the conditions. Okay, so the effects of the short-term and the long-term sleep deprivations are different. So what is shown in this one, for example, this is about the energy allocation. So you see in the short-term deprivation, this is the condition where, your biological functions are, these prevented. So deficits in the biological, you see the deficits in the biological investment. So if we have the prolonged sleep deprivation, there is a, in a way we can call it, there is a reactive homeostasis. However, there will be lots of deficits. And during the recovery sleep, we have lots of investment. So this portion now is enhancing. So because during the recovery sleep, we will have to replenish the deficits, whether in the attention, whether in the other energy allocation processes. Now when there is a acute sleep loss, we all know there is a reduced alertness, there is increased sleepiness, there is increased fatigue, our moods will be disturbed, and there will be difficulty concentrating. So when we look into the neurobehavioral effects of the sleep loss, we can go into the various parameters. We can go for, studying of the, vigilance. We can go for the lapses of attention. There are several methods to do that. And the person with the sleep loss will have the longer reaction times. There will be cognitive slowing. We will make more of the errors. And there will be the working memory failures and impaired decision making and so on. So in the recent years, lots of work has been done at the molecular and the cellular level as to find out what happens when there is an acute sleep deprivation or when there is a chronic sleep deprivation. So since the hippocampus has a major role to play in your working memory, so this is very common model to study the hippocampus. So in this one study which was published in Neuroscience, done in the mice, where the mice were sleep deprived, and they were sleep deprived through gentle handling. That means whenever they go into the sleep, you make them wake up. And another group was kept with the normal sleep. So that was a control set. And they were exposed to the contextual fear conditioning, because when we take the hippocampus, we want to see whether there is a role in the learning and the memory. So they were kept into the new chamber for 3 minutes and given the foot shock for a couple of seconds with some intensity. And they were tested for the contextual memory 24 hours after the training and in the same chamber, and also after 48 hours whether they have a retention or not. So what is shown that for the signaling in the cyclic AMP signaling, this is in the normal cell. So the cyclic AMP signaling, you see it is protein kinase A, and you have this enzyme which is the phosphodiesterase E4, and that will reduce, or that will act on the cyclic AMP, reducing its level. This is in the normal cell which is happening, and the signal go to the nucleus, and we will have the long-term potentiation as a marker for the learning. Okay. In these animals also, after the study was over, the brain was taken, a slice was taken, and LTP was studied whether there was a learning deficit or not. So what was found that in this case, if the animals had a sleep deprivation, there was an increase in the phosphodiesterase activity. So as a result, the levels of the cyclic AMP and the protein kinase will decrease. So also the recent studies have shown that the hippocampal neurogenesis is reduced after the sleep fragmentation. So this has been very interesting topic. In 2000, lots of groups were studying whether the newly formed cells in the dentate gyrus of the hippocampus, if they are made, what are they doing, how are they integrating, if they integrate, are they useful in our learning and the memory or in our emotions. So for that, you can see that the slide of the hippocampus. So it was shown that the new neurons in the adult brain. So in this, again, a study was done on the rat model, because it's very easy to do such studies. It's an invasive study. So the sleep fragmentation, they use the treadmill system. So in the treadmill, you can intermittently stop, and you can turn the system on, so off and on. So that is to have fragmented sleep in those animals. So this was done for 3 seconds on and 30 seconds off. And in the control animals, they had given unrestricted sleep for 150 minutes followed by 15 minutes of the activation of the treadmill. So now again, they used different markers like BRDU and all to see whether they had those neurons, the new neurons, they are still there after the sleep deprivation, and they study the effect on the day 1, day 3, and I think day 6 or 7. So it is found that on the day 1, it doesn't have a much effect on the neurogenesis. However, if it prolongs to 4 or 6 or 7 days, then it affects the neurogenesis of the process. And that was shown as a effect on their memory. So and also it was shown that the hippocampal cell death was observed in those ones. So it affected their long-term potentiation, because that is how this neural process to learn. And then impaired learning and the memory, which we can do through several methods in the rat model. Further, in the humans, so in the humans we can have the psychomotor vigilance test, which is the PVT. So we can sleep deprive a person maybe 24 hours, 48 hours, and it's a very simple test in which in the computer, if you can see on the monitor, we show a Q. In this case, you can see a plus sign Q. So that is nothing but it is attend, it is to focus, and then followed by, we'll give some few milliseconds, another cue, and we'll ask the person to down scroll, and the moment it comes, you stop by or remove it by pressing a button on the monitor. How fast you do will be shown, your reaction time here, and so the person can improvise. So when a person was sleep deprived for 24 hours, so at this PVT test was conducted every 1 hour, starting from 11:30 onwards till 6:30 in the morning, and this test was repeated about 120 as a standard protocol. And what was found that in mean reaction time increased as the sleepiness. In this picture, the right picture, you can see as and on the sleepiness is increasing, the reaction time is also increasing across the night. So that is how we are making, if there is increased reaction time, some of the responses should be immediate. But if you are sleep deprived, the reaction time will be reduced. Further, in this study, they had also placed the EEG and they recorded the evoked potential. So in this, the lighter lines are showing the person's evoked potential in the sleep deprived state. So P300 is a marker for the cognitive attention. So in this case, if you see in the normal person, if this is the state, in the sleep deprived person, you see this is dramatically reduced. Okay. Not only that, in this group, they also studied various energy, or not energy, the frequency bands. So what they found was that in comparison to the normal state, in the sleep deprived state, there was a reduction in the gamma power, which is required for several cognitive functions. And also, if in this see in the right panel, we can see that the spectral delta power. Okay, so this delta power was found to be enhanced in both these conditions. So these are some of the experiments which we can do in the laboratory in order to see the effects of sleep deprivation or different markers of the sleep deprivation. So these are some of the neurophysiological markers. We can have molecular markers and so on. So the sleep deprivation, so since after the sleep deprivation, there is an increase in the reaction time, and that is increasing the subjective sleepiness, increase in the delta and the theta power, reduce P3 amplitude, reduce in the gamma power, and ultimately it's a reduction in your cortical arousal. So that is resulting in the deficits in your attention task performance. So that is a concern. Now also later a couple of studies were done in which acute effects of the sleep loss were studied. So you can see a couple of papers. This is one paper, this is another, this is another. So where the effects of the sleep loss on the various components of the cognitions were studied. And a method called the meta-analysis, so that is done where the reported studies in the where the effects of sleep deprivations are reported, you pick up those studies and then try to see the relation of one parameter with the sleep loss. So these procedures, the meta-analysis, the results of these are all statistical tools. So in this it was shown that the sleep deprivation or the insufficient sleep that leads to the cognitive dysfunctions. There are attentional lapses, impaired working memory, impaired decision making, impaired emotional processing, and that is all leading to the several motor vehicle accidents. So the acute sleep deprivation decreases the brain activation, and it was also found that in the recent years, as Professor Mohan Kumar showed, that they did fMRI studies in the rat in 1980s. You can believe, you can imagine how much effort would have been taken to do those pioneer studies in animals. But these days, lots of fMRI studies are being done in the human beings so that you can see, from one part to the another part how the information is being processed. So these are basically non-invasive tools, but very effective tools. So in this, it was shown that the acute sleep deprivation, based on these meta-analysis, it's shown that the acute sleep deprivation decreases the brain activation in the frontoparietal attentional networks. We have different networks in the brain. So attentional networks, so the more in the frontoparietal networks, and also intraparietal sulcus. And in the salience work, so that is the one I'll show in the next slide, insula and the medial prefrontal cortex. And also there is an increased thalamic activation after the sleep deprivation that may reflect a complex interaction between the de-arousing effect of sleep loss and the arousing effect of the task performance. So this is the one, as sir showed in the beginning, we have a well-defined default mode network. That is the network active. For example, you are sitting silent, you are just thinking about yourself, you are not engaged into any active work. Then your default mode network will be active. When you are doing anything of attention, then your other network will be active. So for that you have now the salience network are coming into action, so whether to take you into this mode or into this mode. So these are the reciprocal modality through the well-defined neuronal group in the brain, which can be detected through the fMRI studies. So in this default mode network, it is primarily the medial, aligned more medially. So you can see the medial prefrontal cortex and the, this cingulate cortex. And whereas the salients are more lateralized, and here also dorsolateral prefrontal cortex, these are more laterally organized. So then looking into what are the neural signatures of the sleep deprivation, or how does it affect the cognition and the emotions. So going further, the effects of the sleep deprivation does not simply reflect the absence of sleep, the benefit attributed to it. Rather, they reflect the consequences of several additional factors, including extended wakefulness. That's what I begin my this talk, that please don't take sleep deprivation as a simple absence of sleep. Many more things are happening. If some of you may have read that long back in 60s, one study was conducted as to how long the person can stay awake. So that person stayed awake, that was a student, about 21 years old. He stayed awake for 11 days continuously, about 260 hours. What was found that they did not have a major deficits. However, that person complained that for years together that he has a feeling of sleepiness. He doesn't feel normal, right? So, sleep deprivation is much more than just the absence of sleep. So now I have 5 minutes. Okay, so in the last 2-3 slides, what I will show is that recently one work was published which says that how the sleep deprived human brain looks. So they took 3 motivational reasons for this. One was a biological perspective, neurobiological perspective. That means we need to characterize that which networks in the human brain are more vulnerable or more resilient to sleep or insufficient sleep or the sleep loss. Or also we need to understand or determine how we have certain comorbid sleep disruption present in several neurological and psychiatric conditions. We have Alzheimer's disease, schizophrenia, anxiety disorder, several addiction disorder. In all these things the sleep is impaired. So there is a need to understand on how these are contributed or result from these disorders. And then from the societal standpoint, such scientific evidence could in debate regarding the sleep recommendation for the public and the professional health policies, because sleep is a dynamic process, as shown in the initial second or third slides, that our requirement of the sleep changes from neonate to the adult age. So once we know that, okay, for the population it may not be a single timings, but yes, a range that will be beneficial for the people, because at times if you are sleep deprived, the person coming to the office may not have the efficiency in the work. So that is a loss to the country in terms of the money, because if the people are there, physical presence doesn't mean anything. People have to work also. So foreign countries, some of the people who are traveling a lot, they have made the nap pod in the office. India, I don't think it will still have the scope. It will take some more years till the time the problem becomes very grave. Now looking next 1 or 2 slides, so the sleep deprivation altering the network integrity. So we have been seeing this kind of a flip-flop model. So you see the thalamus here, you see the fulcrum. So either one stage goes up or one stage goes down. So this is a reciprocal relationship. So in the sleep rested stable state. So when you have normal sleep, so this picture is about when you have the normal sleep, and this is the one when you have the sleep deprivation, how this happens. So how does it affect? So when you have the on-task activity, so your prefrontal networks are engaged. So this one is now default mode and network is now lighter, whereas in the off mode now the default mode networks, they are active. However, in this sleep deprived state, these two states, the alternation or the change, it becomes faulty in comparison to the normal state. So sleep deprivation, that shows that sleep deprivation is affecting these networks, and that is reflecting in their various cognitive, the functions. So what you can see, I had just mentioned in the initial one, so that we have the medial default mode network, which is shown in the blue color picture, and here it is the prefrontal, which is shown in the frontal and in the infraparietal region. Further, so this is the same. Also, if we always, when we look for the motivation, when we look for these attentional networks, recruitment of these attentional networks to do the reward function, then we look for the sleep related, again you see in this picture, what we can see the different types of the receptors. So these are the dopamine the receptors, D1 and D2 and D3 receptors. So in the normal resting state, or the people when they have the normal sleep, in this one there is no bias for the avoidance or the approach behavior. So what does it mean? That means, for example, these dopamine receptors, they will bind more to the D2 and D3 receptors. However, when there is a sleep deprivation, there is a downregulation of these D2 and D3 receptors. As a result, there is a change in the ratio of the D1 to D2, these three, so which you can see here. So there is a change in the bias now. The avoidance behavior is being increased. So that is what we see, that when we are sleep deprived, we see our youngsters, their risk-taking behavior increases, and there is a hypersensitization of the reward system. Because of the downregulation of the D3 and D4 receptors and the increase in the D1 receptors. So that means nothing, but that means there is an inaccurate ability to discriminate between the trials involving, in laboratories we do several tests, Iowa gambling tests, some of these tests are done in order to find out. So the inaccurate ability to discriminate between the trials involving monetary reward and the punishment values, and as well as some trials involving no monetary reward and punishment. So doing this, we are able to predict what happens to these models, whether they become unstable under the sleep deprivation. So the sleep loss, because also our emotionality, yeah, I'll just wind up. So enhanced amygdala reactivity to the negative stimuli. So a normal person, a sleep deprived person will see it as a more negative intent. So there is a reduced activity to in the medial prefrontal cortex and the amygdala connectivity, and there is a defective functioning of the salience detection network, which are important for the reciprocal change into one state default mode network to the prefrontal network. And this is the last slide we have. If we have mostly what were being described was acute sleep loss. When we have the chronic sleep loss, there will be, it will be challenging to, or this will be the risk factors to the cardiovascular diseases, metabolic disorders, immune dysfunctions, obesity, affective disorders, impaired daytime functioning, and so on. So the adequate sleep duration and the quality is essential to maintain the proper metabolic functions, hormonal regulation, and appetite regulation. With this, thank you very much.
Thank you, ma'am. I request Dr. Sushil Keja, Associate Professor, Department of Physiology, Jawaharlal Nehru University, to speak about sleep and memory.
Good afternoon, everyone. Thank you so much, Professor Malik, that you asked me to come and talk about this medical student about sleep and its function about memory. Thank you, Dr. Jindal, for organizing this kind of sleep course. It's really wonderful. I am, as I said, Dr. Sushil Jha. I'm Associate Professor at Jawaharlal Nehru University. And I do research to know what sleep does. So the basic question in the lab that we are asking, does sleep play an instructive role, or the state it just simply allows something to happen? So this is a very crucial question. We are trying to understand what sleep does. But here I will present what Professor Mohan Kumar talked about, the function of sleep, and Dr. Gulia also talked about. I'll just present a little bit of data so that you can understand, and also to expose you how we conduct animal research. So not too many data, but I will try to make it more simple. So, as you all know, that we have a different stages of sleep, and the EEG changes in animal model also. It's like during non-REM sleep, there is high amplitude and low frequency waves, and during wake, the EEG is low amplitude and high frequency, and similar that happens during REM sleep. But whether both the stages has an equal role in memory consolidation, or they play some differential role, that still is not very clear. So we will see how non-REM and REM sleep does to what kind of memory. So in general term, memory is classified into terms of their how we retrieve memory into declarative memory and non-declarative memory. So that is when we put a lot of emphasis on the brain to retrieve data or retrieve the stored information, those kind of memory are categorized into declarative memory. And something that happens is spontaneous. You don't have to put emphasis on your brain to get the information. Those are called non-declarative memory. So what you learn, what you mug up, what you read, all those comes under the declarative memory. And the non-declarative memory, generally the skill, that how we learn to drive car or motorcycle, those are under category of non-declarative memory. So non-declarative memory, as you know, is like you don't have to put a lot of emphasis on brain. I do not know whether we can say that declarative memory consolidation and non-declarative memory consolidation are following a different procedure. Till date, we know all these kind of memory follow similar procedure. But one basic difference is non-declarative memory doesn't take that much of time to retrieve. It means there is a quick retrieval mechanism and no hindrance. To know what sleep does to memory, here I'm just giving you an example of non-declarative memory, and that we call contextual fear conditioning, and this what Dr. Gulia also talked about. So in contextual fear memory, what we do, like you keep the animal in this cage, and this is agreed through that electrical shock is given, mild electrical shock is given. It is not fatal to the animal, but it is a little bit painful, obviously. So how many times we present? Only 3 times. And you see this animal as a normal behavior of the animal inside the cage, little bit fast forward movie. And when animal will get shocked, animal will jump. So these are all normal behavior. Now animal got shocked, jumped. 3 times that kind of shock is given in contextual fear conditioning. And testing is done because here what animal learns, the context. When the animal was kept, the bedding has, the same bedding was used next day for testing. Some fragrance is given, some reminders are given, like next day when you are testing, so animal learns that, okay, in the same context, I experience shock. Now 24 hours later, if you see the behavior of this animal, it's not moving. It will be active a little bit and then again moves. So that is their spontaneous freezing response. Means animal is scared, animal doesn't want to move. And then it is being quantified as freezing as a mark of learning. And here the experiments, the way it is done, this is a baseline what you saw, that is a training when animal was active, and 5 hours of sleep deprivation, manual handling, the way Dr. Gulia explained. And then 18 hours animal was allowed to sleep in their colony, and the next day animal was tested. And you will see this is what the person freezing response. Animal, those were sleep deprived, and the animal, those were non-sleep deprived and stress control. So sleep deprived animal did not learn that the context was so scary. And how long? Only 5 hours, when? Immediately after training. So that is like a timing is important. Someone has shown earlier that if the sleep deprivation will be performed not immediately after training in mice, if it is performed 6 or 7 hours after training, then this impairment doesn't happen. So in animal model, memory consolidation after learning required sleep immediately after training. In human, the night is important. Then, if sleep has certain role in memory consolidation, so 2 ways we can learn. One, we don't allow them to sleep after training. And then another way, another approach is just you record sleep after training and testing and see if there is any change in sleep parameters. So in this case, what has been found, there is a non-REM sleep window. Means non-REM sleep increased after training, but in a particular window that here you can see here the 2 to 3 hours. So the 2 to 3 hours after training, at time particularly non-REM sleep increase, and if you do not allow them to sleep, that augmented non-REM sleep, memory is impaired. So that suggests sleep is important, and at the particular time window, the amount may go up. But as I said, non-REM and REM sleep, are they contributing equally? In this case, unfortunately not. Non-REM sleep is increasing, but REM sleep is decreasing after fear conditioning. And that is surprising. Why REM sleep is decreased? So, certain cases, it has also been found, there's another task called Morris-Watermaid's task, where it has been found that REM sleep is increased after training. But here in this case, REM sleep decrease after training, non-REM sleep increase. So these are the studies that suggest that non-REM and REM sleep are playing important role, but they have a different role in their lifetime. I'm skipping this slide. Now another question is, why do we need sleep for memory consolidation? Memory consolidation is a different mechanism that like different brain areas are involved. Not the sleep area are involved in memory consolidation. So why do we need sleep? So before that, we have to understand how memory consolidation is taking place. So the simple example is about memory consolidation. If something is learned, some neural changes takes place. And at the cellular level, what happens, as Dr. Gulia showed, there are second messenger system, protein kinase, their level goes up. There are certain genes, their level goes up. So molecular events are taking place. And to induce these changes, glutamatergic receptor, NMDA receptor plays important role. So when neurotransmitters are released after acquisition, that glutamate activates NMDA receptor, and NMDA receptor in turn activate protein kinase A, their second messenger system, that ultimately express gene. This is called one-time phenomena. But it has been found this one-time phenomena is just to induce the process for memory consolidation. And memory consolidation takes hours. All these mechanisms that we are talking about, these are takes hardly half an hour to induce. But memory consolidation takes 4-5 hours or several days even. So what is like, this cannot be a single cascade inducing effect. So then another theory was proposed that is called synaptic re-entry reinforcement theory. So in this, what happens, like first time during acquisition, if the system is activated, then it requires reactivation after a certain period of time. So multiple times the system is reactivated, and reactivation is important for memory consolidation. And it has been shown that the neurons, those are playing important role in hippocampus or prefrontal cortex during acquisition. Their activity goes up, and the same activity is replayed during sleep. So the neuronal population, those are involved in acquisition, when they are activated at the time of acquisition, the same populations get reactivated during sleep in a similar pattern. This has been shown by this group. And this here you can see another example of synaptic plasticity. This is called ocular dominance plasticity. So this was the firing rate of a neuron at the baseline. And when synaptic plasticity was induced, now you can compare how the neuronal firing increased during non-REM sleep and REM sleep. So the neuronal firing increases during sleep, and that is why we need sleep for memory consolidation. There are several events that take place. I will take to you those events one by one. Another, what Dr. Gulia has shown about this homeostatic sleep drive. where delta waves, theta waves are associated with non-REM and REM sleep. If someone is sleep deprived, it gets augmented during recovery sleep. And that is what is suggested. It was suggested that sleep is homeostatically controlled. That not only happens after sleep deprivation during recovery sleep, but it also happens when animal learns. So after learning, when animal is going into sleep, those electrophysiological markers in the brain waves are also augmented. For example, here you see after learning, that is learning and the testing, training and testing days, this delta waves and the sigma waves, that is 0 to 2 hours after training and testing, 2 to 4 hours, you see how the delta is increased and then going back to the normal level after 6 hours. And the sigma waves are still up. We haven't done, like it is not known after how long it is going to come back at the basal level, but you see after training and testing, the waves are very high. At the cortical, during non-REM sleep and REM sleep, this is a theta waves, and this is at the cortical, subcortical areas. So these electrical signatures are definitely augmented after learning, and that too particularly during sleep, non-REM and REM sleep. And these electrical signatures have certain roles to trigger those neuronal activity. That is what it is believed, and that is how the neuronal activity is also augmented during memory consolidation phase. So next, how does sleep helps in memory consolidation? That is a very logical question, right? So gene is highly expressed after learning, and it is also expression increased during sleep. And here there are certain examples, the 4 memory candidate genes, that is FMN, GSK, NCDN, and SHANK. You see after training, the level is up at 1 hour, then came back to the normal level, up at first hour, then it's gone down. So certain genes, their expression is increased after training, and especially at 1 hour or 2 hours after sleep. And if using RNAi inhibitory, like RNAi interfering mechanism, if you interfere the expression of these RNAs, then their memory is impaired. So that suggests that this expression of gene that increases particularly during sleep, and that helps in memory consolidation. Not only that, there are 2 brain areas where even in the adult brain, neurons are always regenerating. And those 2 areas, what even Professor Mohan Kumar has talked about, dentate gyrus and as well as the subventricular zone. In dentate gyrus of the hippocampus, the neurons, regenerating neurons, are more after hippocampal dependent training. So if you give hippocampal dependent learning tasks, and you see how the number of neurons, these all dotted neurons, you see the numbers are more in a trained condition compared to the control. But after sleep deprivation, the increased number has gone down to the basal level. So after learning, if the neurons are dividing and animal is not getting proper sleep, these dividing neurons are not at the level, but rather it remains at the basal level. So that is another way to show that it may contribute in memory deficit if sleep is not achieved properly. The last one, when does it happen? So day and night, we have like a human, they are active during daytime and we go on bed at the night, and that too only for 6, 7, 8 hours. But the animal is the other way around, like rats are sleeping more during daytime and they are active more during nighttime. So this experiment was during daytime when they sleep more. So 6 hours sleep deprivation or 5 hours sleep deprivation after training causes memory deficit. But another group have shown, this Merlo group, that if you do the same experiment during night, the 6-hour sleep deprivation is not contributing in inducing memory deficit, rather 12 hours is sufficient. 6 hours is not sufficient. So here the result is in the rat when they learn better, during daytime or nighttime. So active phase is the night and the inactive phase is day. So there is a difference. Animal learns better during night than during daytime. If that is the case, then when you ask like nighttime, you need 12-hour deprivation where the daytime only 6 hours. But what if we combine sleep deprivation and circadian alteration together? What will be the impact? So this is what the experiment has been done. So one group where no sleep deprivation, they were kept, like this is the 12 hours dark, 12 hours day. So this is a night experiment. So 12 hours dark, this control animal. Then another group is they were non-sleep deprived, but 6 hours, there was a phase delay. So instead of keeping them in 12 hours dark, 6 hour light, 6 hour dark, there's a stress control, same pattern. And sleep deprived means during the light, you keep animals sleep deprived. You do not allow to go into sleep after training. Again, the contextual. And the test after 24 hours. The result is none of these animals in 6 hours showed any change. They showed proper learning. But the animal, those were sleep deprived and their circadian cyclicity was also altered. Now combining 6 hour sleep deprivation with circadian alteration, they are not showing proper learning. So their learning was impaired. These are the story of sleep deprivation and learning. So now another question is if we are living in the world where we can't change our lifestyle. So if we have to perform, we have to perform. We have to be awake, we have to be awake. So is there any way that we can reduce the sleep deprivation mediated detrimental effect on learning? So yes, there are ways to affect that, and one way is through epigenetic model. In epigenetic model, so in epigenetic model, what is there, like DNA remains tightly wrapped with histone proteins, and when they are tightly packed, then gene will not be expressed. And when the acetyl group is bound at that certain specific site, then the tightness is loosened, and then this DNA is unfolded, and then gene is expressed. So the mechanism at the histone level, when it is controlled, gene expression, that is called epigenetic model. And here the two more important enzymes are working. One is HAT, this histone acetyl transferase. What it does, it transfers acetyl group to the different site, and then it opens up the DNA. And another is histone deacetylase that removes acetyl group, and then it packs it. So the gene expression will not take place. So here, there are certain drugs that can be used to inhibit the activity of histone deacetylase, or there are certain drugs that you can induce the activity of this histone acetyl transferase. So here in this case, the example is SAHA, that is quite widely used, this drug for cancer treatment and also. So here, what it suggests, if the SAHA is given into the hippocampus, where these kind of plastic changes are taking place in hippocampal dependent tasks of the contextual fear conditioning, in the sleep deprived animal, so you see this vehicle group in the sleep deprived, and the PD means that is circadian cycle is also altered. So those animals did not show learning, but the SAHA treated animals showed recovery in their memory. So their sleep dependent memory deficit was rescued in the presence of SAHA. And this is what at the cellular level you see what happens at the level of histone deacetylase level So this is like the normal level, it increased, in sleep deprived it decreased, and the Saha treated level it also increases. So this is how sleep deprivation mediated effects can be rescued. So to conclude, the consolidation of memory requires sleep, and it suggests that the both non-REM and REM sleep has a differential role, and it affects at the neurogenesis as well as at the protein level, and altered circadian cyclicity along with sleep deprivation has more detrimental effect rather than only sleep deprivation, that too. Okay, thank you so much.
Thank you, sir. Moving on to Module 2, Taxonomy and Sleep-Related Disorders, I request Dr. Karuna Datta, Professor and HOD, Department of Physiology, ACMS Delhi, to talk on classification of sleep disorders.
Well, I am very, very glad that the organizers have told me that you can introduce yourself, and this is all that my introduction is. There's nothing much more than this. And I come from AFMC Pune, and I can see a lot of undergraduates sitting behind. So can I see a show of hands as to how many of you actually know what are the different types more than insomnia and OSA as sleep disorders? Can I see a show of hands? Anybody knows anything beyond insomnia and OSA? Very good. So I don't see any hand up. So that means this class is perfect at the right time. So I'll not let you sleep, first of all, at the back. Are you all with me? Right? So this is your first basic class to sleep medicine. Now the second question to you is, how many of you actually get patients where they come and say that we have a sleep problem? Can I see a show of hands again? Very good. So I do see some hands. That means you are seeing these patients who come to you and say that we're having a problem, but somehow we are not being taught, so we don't know, and that's what this course is all about, right? So with this, let me start with a basic slide. We've all been hearing since morning that we have wake and sleep. Sleep is divided into two, that is non-REM and REM. And as we heard today in the morning, Dr. Mohan Kumar gave us a very good insight into a 2019 paper where different states can be identified of sleep using fMRI. But predominantly, you have non-REM and REM. And non-REM is around 80% of the night. REM is 20% of the night, right? Is that clear? Right. Now the disorders which will happen generally happen when it shifts from one phase to another. So when wake to non-REM, non-REM to REM, or essentially something which happens in non-REM, or something which happens in REM, or something happens that in your wake state you are sleepy. So these are the three things that you might find broadly. Now things which will cause you to be sleepy in daytime can be as a result of either a bad sleep quality or a bad sleep quantity. Basics. Is it clear? Yes, so I do see some head shaking. So broadly, these are the symptoms. You get sleepiness. The person will say, I keep on waking at night. There are abnormal behavior or movements in sleep, abnormal physiology or anatomy of upper airway, where the person will say that I have some choking when I get up in the morning, or I have been told that my teeth are not aligned very well, like that. And there may be some central causes which you have to identify. Then there may be problem in stage transitions, which again you will identify. And last but not least, which has been so rightly brought out in the morning sessions, the circadian problems. So the first, and I'm going to talk about how these disorders can actually be picked up, or when do we call this disorder a disorder. So what is the chronic insomnia disorder? Anybody? Anybody at the back? I warned you during tea time, I'll not let you sleep. So anybody at the back, when do you call it chronic? Yes? Okay, so let's go through these slides and the questions will be after the class, right? Okay, so it's the patient criteria, the diagnostic criteria is that the patient reports, so the patient's parent or caregiver should observe one or more following of the following. That is, either there is difficulty in initiation of sleep, maintaining sleep, waking up earlier than desired. There is a resistance to go to sleep. You find these small kids, when you say it is sleep time, they will start playing games, they will go somewhere else, they'll hide behind the bed. That resistance also should be taken into consideration. And last but not least, where they require a parent or caregiver to actually make them go off to sleep. So all these 5 things, you have to see if one or more of them are available. And then the diagnostic criteria would be that the person should have nighttime sleep difficulty, along with it some daytime problems like fatigue, attention concentration, memory problems, as has been rightly brought out by Dr. Jha in the morning, impaired social family, occupational or academic performance, mood disturbances, daytime sleepiness, behavioral problems. You will find this boy in the school who's picking up fights with everybody, fighting, sleeping in the class, not getting good grades, area of concern. If the mother comes to you with this child, you must ask history of sleep problem. And that is a problem which generally is picked up once the child actually gets psychiatric complaints, rather than when we should pick it up much earlier than actually the child suffers. Then you can have proneness for accidents, concerns about or dissatisfaction with sleep. Now one more thing that you must remember, how many of you are sleeping more than 6 hours of sleep? Anybody sleeping more than 6 hours per sleep? Oh, I see some hands here. Good. How many of you are sleeping less than 6 hours per sleep? I must say, very brave. So, if you are sleeping less than 6 hours per sleep, that means you go off to bed. I'm giving you an example at the back. I want you to follow, otherwise you will not follow the entire course. So, if a person says, I go off to sleep at 12 in the night, and I wake up. I go to bed at 12 and I wake up at 5:36 in the morning. Is it insomnia? Is it insomnia there? The 2 girls who are discussing, yes? Yes or no? Can't hear. Yes or no? Is it insomnia if somebody sleeps at 12 in the night and gets up at 5:36 in the morning? 4 people there. Is it insomnia? No? Then what is it? Good. I hear that, like that one person doing this. Remember, it is not insomnia. Why? Because you have not given adequate opportunity to yourself to sleep. Insomnia is when a person is lying in the bed to sleep. Typical history, I go off to bed at 10 o'clock in the night to sleep. I can't sleep. I'm tossing and turning, trying to sleep. It takes me almost 2 hours to sleep while lying in bed. I don't wake up also. I'm lying in the bed only trying to sleep. I sleep by 12. I get up in the morning at 6 and I'm out of bed. This is insomnia. And this insomnia is an initiation insomnia. Have you understood the difference? So this is insomnia where the person has gone off to bed at the right time, given himself opportunity for 8 hours to 9 hours, 8.5 hours, but he's not able to sleep. Now you find a person who comes to you and say that I sleep at 10 o'clock, I go to the bed at 10 o'clock, I sleep in around 10 minutes, and then I wake up sometime at night. I'm not able to sleep after that for 1.5, 2 hours. And once I go off to sleep, now I get up at 6 o'clock. Is this insomnia? Again, I want you to answer. I know you're following me. Is it insomnia? He's gone off to bed at 10. He sleeps within 10 minutes. And now he says, I fall asleep. I wake up sometime in the middle of the night. I don't know when, but it takes me a long time to sleep, maybe 1.5 hours. And then when I sleep, I get up only at 6 in the morning. Is it insomnia? Yes? Yes, so it is insomnia because a person has given adequate opportunity to sleep. Always remember this word. Given adequate opportunity to sleep, not able to sleep, insomnia. But not given adequate opportunity to sleep. So if there is inadequate opportunity to sleep, like you slept at 12 and you got up at 6, it is not insomnia, but it is sleep deprivation. Clear? Right. So in chronic insomnia, this should not be explained. You can only call it chronic insomnia when it cannot be explained by inadequate opportunity to sleep. Right? Okay. And it should be at least 3 times a week. And it should be for at least 3 months. And as we keep on hearing this, don't think everything is only insomnia. There are many things around, many diseases which lead to insomnia, which may be a cause of insomnia. We don't know which one came first. But whenever you get a case like this, who says that I'm not able to sleep, despite giving opportunity to sleep, it's been 6, 7 months, doctor, now I don't know why it is happening. Look for other causes too. It is insomnia, true, but there may be some other comorbidities. What can be the other comorbidities? Take cardiovascular history. Hypertension and insomnia are common. Take diabetes. You must ask. You must even get HbA1c done because you might have diabetes and insomnia linked. They all go as baggage, With insomnia, you can have depression, very common. In depressives and anxiety people, you will find that they have very difficult type of sleep, very different type of sleep. So you should not miss out other comorbidities which may be coexistent. Another very, very common thing is GERD. A person has reflux, he gets up at night, now not able to sleep. So he will report to you with insomnia. He might also say a little bit khatta something I have here, and then I'm not able to sleep. So look for other comorbidities. Now, a chronic insomnia disorder is identified by these 3 things. But what I'm trying to say is, it can be chronic insomnia and something. It can be chronic insomnia and another something. ICSD-2, which is International Classification of Sleep Disorders, earlier used to classify it as primary and secondary. We have given away with that. We don't believe in it. Now, as per the ICSD-3, the International Classification of Sleep Disorders calls it as insomnia because whether it came primarily or secondarily doesn't matter. It has to be treated as insomnia. And that is why it deserves the special name as insomnia. No primary, secondary. So chronic insomnia is if he's not able to sleep, he has excessive daytime sleepiness, or he has any other daytime symptoms as we have just discussed. It is at least 3 times a week, it is at least for 3 months, and it cannot be explained by inadequate opportunity to sleep. Now another common, or rather not common, but you usually see, you might see some videos tomorrow, is narcolepsy. We often talk about this disorder, and these patients often come with excessive daytime sleepiness, and I would say they are commonly missed. They are not common disorders, but these are the disorders which are commonly missed. Now what happens in this disorder is, that the history would be that they have this excessive need to sleep. They want to sleep. There is daytime lapse. You will find them sleeping anywhere and they'll suddenly drop. They laugh, they drop. They have an irresistible need to sleep. And with that, you might find cataplexy. What is cataplexy? This is defined as an essential feature, and the presence of either of cataplexy or of hypocretin deficiency is mandatory. Now, CSF hypocretin deficiency, to call it as a deficiency, you have to do hypocretin test. I don't think many labs in India are doing hypocretin tests. That is why you must know your clinical symptoms and you must be able to diagnose these patients early. So we'll just go through this definition. The presence of one or both of the following is essential, which is cataplexy as defined under essential criteria. Shall I wait? We have some new entrants. I'll just wait for you to settle down. Some 30-40 people have got added at the back. Good. Right. You can settle down here in the middle rows. You can come forward if you want. Okay, so cataplexy is mean sleep latency of less than 8 minutes. Are you following? The girls there? Okay. So at the back, new entrants, please follow from here. So mean sleep latency of less than 8 minutes and 2 or more sleep onset REM periods on a MSLT. Now what is MSLT? It is a multiple sleep latency test which you do after polysomnography, and in the morning you give an opportunity for the person to sleep while he's all hooked up in the lab. I'm telling you in a very, very broad way, more will follow. And if a person sleeps in that lab in less than 8 minutes and has 2 or more REM sleep onsets. When you sleep in the night, which is the sleep that you start with, non-REM or REM? Very good. So you start with non-REM. Now this person in the morning actually starts and goes off to REM. So you get 2 of these episodes where he starts with REM. Mean sleep latency is less than 8 minutes, and on an MSLT via standard technique, you find sleep onset REM periods within 15 minutes of sleep onset after a nocturnal polysomnography which has been done, criteria achieved, right? The second criteria is the hypocretin concentrations less than 110 picograms per ml. Now in young children, sometimes you find narcolepsy can be present as excessively long night sleep or as resumption of previous discontinued daytime napping. And sometimes it can be also suspected narcolepsy type 1 when clinically, but the MSLT criteria are not met and possible repeat is what you should be doing. Now type 2 is in which cataplexy is absent. You find a mean sleep latency of less than 8 minutes. You find irrepressible sleep need. You may not find cataplexy, and you find that the hypocretin 1 concentrations are reduced and there is hyposomnolescence which cannot be explained. Again, this is one place you have to be careful. These are often missed. I think the sessions will follow where we'll talk about it in detail. But remember, these patients come with excessive daytime sleepiness. Now this patient will also come with excessive daytime sleepiness. These are the patients you will find in the class sleeping, not able to concentrate. But what is the problem with them? They have not slept at night. They are the ones who have not given adequate opportunity at night to sleep. They are the insufficient sleep syndrome or the sleep deprivation patients. Here, again, there will be an irrepressible need to sleep. The person will just not be able to follow. He'll just go off to sleep. But you will find that the pattern is almost for 3 months. The resolution of symptoms occurs with sleep. The moment they sleep, they're fine and fresh. And the problem is that they have not given adequate opportunity for themselves to sleep. How do you log it? I ask somebody, do you sleep fine? They will say yes. How many hours do you sleep? I sleep 7 to 8 hours. But how do I come to know he's actually sleeping 7 to 8 hours? More are coming. I think some class has got finished. Is it? 50 odd maybe. Good. So how do you know that the person is actually not sleeping? Can you measure it? Anybody? You come to me and say that I am sleeping fine, but I really want to sleep during daytime. So, how do I come to know that what you're saying is fine? What can I do? Can you do actigraphy? So, you can do actigraphy and you can ask them to fill in a sleep diary. Okay, if you fill in the diary, you can write down what time did you sleep, what time did you wake up, you can come to know how he is sleeping. So this is something which you should do to find out, preferably for 2 weeks, that how their sleep is. Now a word about parasomnias. What are parasomnias? They can be non-REM or they can be REM. These are abnormal movements which happen. In non-REM, you have these disorders of arousal. Suddenly, the person in non-REM sleep will just wake up with, there may be confusional arousals where he'll wake up. There can be sleepwalking, sleep terrors, and sleep-related eating disorders. Whereas, and there is a criteria. Now, non-REM is, I will not show this. So, when the person starts to sleep, who all was seeing the first slides which Dr. Kumar was telling you, you must have seen that how the sleep cycle went across, right? You remember the patient went off to sleep and the first thing he went into was with sleep? Non-REM. And then what happened? After around 90 to 110 minutes, one cycle is 90 to 110 minutes, and in that cycle, just at the end, you get REM sleep. Then again, he, comes back to end to, or maybe awake also, a little bit arousal, and then again he goes into non-REM, then REM, then again non-REM, REM. As the night progresses, your REM length keeps on increasing, right? So if it has to be a non-REM problem essentially, when would you see it? Towards early morning or towards earlier in the night? Right. So you'll see it towards earlier phase in the night, and that's what is the criteria. You must remember that it should occur during the first third of the major sleep episode. So that hints you to a non-REM associated problem. And what are the other things? There can be inappropriate or absent responsiveness to efforts. It can be a limited or no associated cognition. You wake him up, he will not come to know what's happening. And the disturbance is not explained by other disorders. Then is the REM related parasomnias, which is REM sleep behavior disorder. The person will be doing dream enactment, he will be actually doing, you've seen that movie? There are many movies in which you have these REM parasomnias. You can have nightmare disorder. The person will suddenly wake up after seeing a nightmare. And what happens in this is there are vocalizations, the person is talking. You will find that the polysomnography will see that there is a REM sleep with atonia, and during that these things are happening. Also you will find that sometimes they will be acting out one's dream. Somebody kicks, you wake him up, you say, oh, I was playing football in my dream. And plus it will not be in the initial 1/3 of the night because REM is more towards the latter part. So you will find it will be little more later in the night. Restless leg syndromes, it just follows. So maybe I will just hurry this up. This is another one cause where the patient will say, I'm not feeling fresh the next day. And this session is just after this session, so maybe I'll just rush through this. Just letting you know that there is an urge to move the legs. The person keeps on saying that I want to move the legs, and it is usually accompanied with very unpleasant sensations in the legs. The features cannot be explained by other things, and generally it happens towards the evening or the night time. Periodic limb movement disorder, remember the frequency, more than 5 per hour in children or more than 15 per hour in adults in a polysomnography with evidence. So that's when you will call this as a periodic limb movement disorder. And last but not least at all, which is the most important for all of you there, is the circadian rhythm disorders. Now, why circadian rhythm disorders are very important? Because we love to chat at night on WhatsApp and attend the class default mode, sleep mode during the daytime. So what you enter up is, you're ruining your circadian rhythm. And what happens is, Sleep deprivation. Sleep deprivation is not circadian rhythm that you have done to yourself because you're not able to sleep in the morning. But what can happen when you start doing this delay in sleeping? You can end up in having a delayed sleep phase syndrome where you tend to sleep late in the morning if given a chance. Now that's the time you should worry, not delay, in fact take help. You should not have a circadian rhythm disorder. Your performance will deteriorate. Your academic performance will deteriorate. Learning and memory will deteriorate because no college will start at 12 o'clock for you. They will all start at 8 o'clock, 9 o'clock for you. So you will end up depriving yourself. Another thing seen in elderly population is advanced sleep phase syndrome. You can have, somebody can come to you with irregular sleep phase, no time to sleep, no time to wake up. Non-24-hour sleep-wake cycles, shift work disorder, so commonly seen, jet lag disorders, most of us must have seen when we do transmeridian travel. And last but not least is one big dabba which is not otherwise specified. Now the general criteria is that in this you should have a chronic pattern where the rhythm is affected. In fact, in sports we had this person, he had gone on an international sports event, and when he came back for 3 months, his entire pattern was topsy-turvy. Why? Because he was entrained for that event to play abroad. When he came back, he had to be set back. But he did not understand the importance. He came to us after 3 months saying that I'm not able to do it myself, now you help me. So remember, it's very important after a transmeridian flight, somebody has stayed abroad, done some part, now comes back, is not able to get back in 2 to 3 weeks, must help, otherwise he'll end up in a disorder. So the take-home messages are very simple. Look for important symptoms, take appropriate history, complete relevant questionnaires. For each disease we have validated questionnaires now, it's all available on net. Conduct a polysomnography as per protocol, MSLT in case it is required, and take the case as patient as a whole. Ask for other histories of hypertension and other comorbidities which may be coexistent. Thank you.
Thank you, ma'am. Now I request Dr. Anjana Talwar to talk on what goes wrong in obstructive sleep apnea, pathophysiology and risk factors.
So that we can read it on the side the next. Is this fine? Yeah, this is okay. Please come. Okay. So there is a, this is the pointer. So the slide is from here on. I'll just take it out. Slides also you can change from here. From here? Or otherwise from here only? You can use the other pointer.
Yeah. Good afternoon, all of you. So today now we are having pathophysiology of obstructive sleep apnea. This is a disorder which many of us are aware of. Most common condition associated with sleep and breathing. I am thankful to the organizing team and the Indian Society for Sleep Medicine for allowing me to take this lecture. I am working as a professor in Department of Physiology in All India Institute of Medical Sciences, New Delhi, and having a respiratory research lab. So to start with, as I said, this is a condition very common. We all experience and see many cases where a person reports, a person reports with the history of their spouses having snoring at night. So in this case, the female reported to an ENT person with a history of ear pain and was diagnosed with otitis externa due to trauma due to earplugs. She had been using earplugs because the husband is having snoring throughout the night and she can't sleep. And to further give the history, they came to know that he was a fat person having a very big neck circumference, 18 collar size. So this is the way many of the cases report, though it is considered to be a very common and easily recognized condition. And because of its association with other conditions, it is a very dangerous condition also, because it is a dangerous scenario because it is a treatable condition and many of the cases go unrecognized because of this. We have been hearing earlier, Dr. Malik had given a lecture on regarding the respiratory changes in sleep. There is a change or a decrease in the ventilation which occurs with sleep. And this decrease in the ventilation is to the tune of 0.5 to 1.5 liters. And this is because of the decrease in the metabolic rate, decreased chemosensitivity, increased airway resistance and wakefulness. As a result of this, the changes in PaCO2 and the oxygen are to the tune of 3 to 10 for oxygen and CO2 with 2 to 8 millimeter of mercury, and the decrease in saturation by 2%. Now, whenever the breathing is disordered in sleep, it is called sleep disordered breathing. And the spectrum of the sleep disordered breathing includes snoring, upper airway resistance syndrome, hypopnea and apnea, that is the obstructive sleep apnea, followed by obesity, hypoventilation syndrome. All of these conditions, except for the snoring, are associated with repeated arousals throughout the night. Most common of these conditions is the obstructive sleep apnea, of which we are going to talk about now. Before we do that, we must know what is the meaning of apnea and hypopnea, which is being used repeatedly for obstructive sleep apnea. Apnea is the cessation of inflow for more than 10 seconds. 10 seconds or more when there is absence of airflow, it is called apnea. Hypopnea by the American Society of Sleep Medicine has given that decrease in the airflow for more than 10 seconds of more than 30% of the flow and associated with the greater than 3% desaturation and or arousal. This is one criteria. The other criteria does not include arousal for the diagnosis. Now, the apnea patterns which are generally seen are of three types: obstructive, mixed and the central. If we consider airflow and the respiratory effort, we can see there is an absence of airflow in all the three, whereas the respiratory effort is the one which helps us in distinguishing the three types of apnea. For the obstructive sleep apnea, there is presence of ventilatory effort or the respiratory effort, whereas in cases of central apnea, there is absence of ventilatory effort. Whereas a mixed apnea is a combination of two. In the initial component, there is central apnea followed by an obstructive component or increased effort. Now, coming to the obstructive hypopnea, where there is recording of EEG, airflow and the effort from the rib cage and the abdomen, and in addition to it, the saturation. So obstructive sleep apnea, as we see, there is a decrease in the airflow, and this is then followed by more, it is for more than 10 seconds, and this is then followed by arousal and a decrease in the saturation as compared to the baseline. Obstructive apnea, on the other hand, has got a complete absence of the airflow associated with increase in the respiratory effort. At the end of the apnea, there is arousal, and this is followed by a decrease in the oxygen saturation. Central apnea, we have talked earlier, again same thing is there. There is absence of airflow recorded, followed by arousal later when the apnea ceases, but there is complete absence of the effort which is seen in this condition. And this is also associated with a decrease in the hemoglobin saturation. A mixed apnea is a combination of the two. In the initial part, there is no respiratory effort as seen here. This is then followed by a portion where at this part the person is sleeping, having no airflow, the apnea is there, but effort continues, and again associated with decreased saturation. Now, as I said, this is an important condition and treatable. The prevalence of sleep apnea in 30 to 60%, this is from a US population, it was reported to be more than 9%. That is, here if you see, this is the recording of AHI more than 5, and here it is the sleep apnea syndrome, can't see from here. This is the asthma recording just for the reference. So if we see here, these values which are shown here are 4% in men and 2% in women. I can't actually read from there. I hope you can all read, but those at the back, I'm sure they can't make out. There are not many studies available regarding the prevalence of obstructive sleep apnea in India. This is the landmark study which was given, which was done in Delhi. This is by Dr. Sharma et al. And was published in Chest. This reported the obstructive sleep apnea overall prevalence to be 13.7 and the prevalence in males and females to be 4.9% and 2.1% respectively. Now, what actually goes wrong in the obstructive sleep apnea? What we have here is the sagittal section. Just for the reference, it will show the area of concern to us. To orient here, we have nose, maxilla, and this is then soft palate and the uvula. Tongue is shown here, and at the back we can see pharynx. Now, the portion of the airway which is there between the corona and this pharynx, this portion oropharynx, this is the part of concern, and most of the times the airways will collapse, or the collapsing of the upper airways occur in this part when the person sleeps. This part becomes important because this area there is no rigid bony support and it is collapsible. It is required for the speech and for the swallowing, but then again it becomes important during sleep because then it gives a greater duration, it allows the greater duration where the airway may collapse. The normal shape of the airway in a healthy and a person with obstructive sleep apnea is entirely different. This is a normal shape. In a snorer, round, and as the disease progresses and apnea occurs, it goes on to a decrease in the lateral diameter here. Now, this is a record showing what happens in a person, healthy individual and an OSA patient when he is awake or during sleep. This is the recording of the genioglossus muscle, which is the main muscle of the dilator of the airways, and the tensor palatini, which is connected with the palate function, the soft palate. So, if we see that in a normal healthy subject, during a wake state, this is the genioglossus MEMG, and this is the one in non-REM, and the airflow is maintained whether the person is awake or in sleep. There may be slight difference but not much, There is a decrease in the EMG for the tensor palatini, but not for the genioglossus, much changes there. Whereas when we have a OSA patient, he has a anatomical abnormality or a tendency to have a collapse. The collapsibility is possible due to certain features which are present in these patients, which we will talk about later. But as a result of that, to maintain the airway in the open state or the dilated state, in the awake condition only, the genioglossus has showing increased activity, and as the tone decreases during sleep, the EMG recorded is decreased. Whereas the tensor palatini muscle also shows a decrease in the tone, but during sleep, the flow decreases tremendously. Here it is complete apnea which is shown. There are different factors which we have to consider when we are going to see what happens in OSA. It could be related, there may be structural factors, neurological factors, and certain others which do not go into this category. OSA patients generally have excess of upper airway soft tissue. Their caliber is generally smaller even during wakefulness. They have larger tongues and longer soft palates, not all of them. Someone may have one problem and the other may have another problem, like larger tongue may not be there for everyone. So they have a longer upper airway, and in them there is a decrease in the lateral diameter, as I had shown in the earlier slide, where the configuration changes. Now various structural factors which are related to the upper airway anatomy and become important: macroglossia, that is bigger tongue, lateral peritonsillar narrowing, enlarged soft palate, tonsillar hypertrophy, deviated nasal septum, retrognathia and micrognathia, and the narrow hard palate. So these are the conditions which will predispose an individual to the narrowing when he is going to sleep or when he sleeps. There are neurological factors, that is the neural modulation of the pharyngeal muscle activity. We have the local inputs, that is negative intraluminal pressure, the respiration related inputs from the center, and there are other factors like endocrine disorders, hypothyroidism and acromegaly, specific genetic conditions, and drugs such as alcohol and sedatives which could be important. Now, one thing we have talked about, that there is in these patients there is an anatomically compromised or collapsible upper airway due to various conditions. This can be determined by their critical closing pressure or the pressure at which the airways will collapse when he lies down. In addition to it, the three other factors which are responsible for different phenotypic traits are the responsiveness of the muscle, that is genioglossus, and the second is the arousal threshold. These individuals will wake up prematurely to apnea. Then in addition, there is an oversensitive ventilatory control system, that is they have a high loop gain. So what is the concept of this critical closing pressure? This is the figure on the top. It shows the airway, whereas this is the pressure upstream and this is the pressure downstream. Here we have the critical pressure outside in the collapsible portion, and as a result of this pressure, certain resistance is being offered by these two components. So in between we have the collapsible component. Now when the critical pressure here is more than the pressure which is upstream or the downstream, there will be total collapse of the airway. On the other hand, if the upstream pressure which is there in the airway is more than the critical pressure, but which is again more than downstream, there is a flow, and towards the end of the portion there is a closing, and this is the one which is being affected. Like supposing the downstream airflow, the pressure is less, there will be closer, but then the upstream area, as the pressure there gets increase due to the incoming air, it overpowers the critical pressure here and it opens and the patency is maintained. So there are times of opening and closing at this point. Whereas it will be totally patent if the pressure in the upstream and the downstream is always more than the critical pressure. So how can we determine this critical pressure, closing pressure, or the collapsibility of the airways is by lowering the CPAP pressure and recording at what pressure there is a collapse of the airways. This is the way it is done, and it has been shown in this diagram that in this study, that is the normal individuals as compared to the sleep apnea patients, you will see the pressure difference which is there. The critical pressure is elevated in sleep apnea patients compared to the normal control. Whereas in this condition, on the other hand, it shows the different groups: normal healthy individuals, snorers with upper airway resistance and with obstructive sleep apnea, and all of them, that is as compared to healthy, have a higher critical threshold, the higher value. Now, this value is more in snoring but still higher in the patients with sleep apnea. So if you see this, this is a record showing the relation between the scatter plot between the critical pressure as well as the AHI index, that as the AHI index increases, there is an increase in the closing pressure for the collapsible airway. In this study, they had divided the patients according to the normal division, which is done into 4 groups: control subjects having a pressure of less than -5 centimeter, whereas in between -5 and -2 there is overlap, and beyond that were the patients with obstructive sleep apnea. Though this is again showing now coming to the next, as they were lowering the pressure from the CPAP, they recorded the responsiveness of the genioglossus muscles. This is the muscle responsiveness, that is percentage of the maximum EFG by the epiglottis pressure centimeter of water, and it shows the relation that the responsiveness on the obstructive sleep apnea patient was less as compared to this. Now, in these conditions, you will see there is certain overlap between the 2 groups, whether it is for the genioglossus responsiveness or in the next pathophysiological feature regarding the arousal threshold. There is always some overlap which is seen between the 2 groups. The arousal threshold also was seen, and the arousal threshold was considered to be greater when it was seen to be more than -15 centimeter of water of negative epiglottic pressure. Again, there is an overlap between the 2 groups, but the values are much lower or more negative actually for the obstructive sleep apnea group. Then coming to the next concept, that is the loop gain, that is the loop gain in these individuals, that is obstructive sleep apnea, was measured. How do we measure? We give a disturbance. There is a ventilatory disturbance followed by the recording. As we are recording, what change is there or how many oscillations are there in the system as a result of the changes brought about by changes in the pressure will help us in determining what is the loop gain for the individuals. In these individuals, the patient group had got the values which were much less. The loop gain was to the order more, that is from -5 to -12. More of the patients were in this category. So now coming to the overall picture, that these individuals had a narrow or collapsible upper airway, and in addition they had low arousal threshold. Many of them had poor muscle responsiveness, and in addition the high loop gain. Now the arousal which occurs in these patients at the end of the apnea is recorded here. That is, as the apnea progresses or event progresses, there is a built-up or the difference between the ventilatory demand of the individual and the oxygen supply or the airflow which is coming, which when reaches a threshold leads to arousal in the individual. Now, coming to the whole cycle of pathophysiology of sleep apnea, then when the person is awake, that is having categorically a smaller airway or some predisposition with some neuromuscular compensation going on in awake state. With the sleep, there is loss of this neuromuscular compensation as the muscles relax, and there is decreased pharyngeal muscle activity, airways collapse, leading on to apnea, hypoxia and hypercapnia. This is then followed by, in association, there is increased ventilatory effort and arousal from sleep occurs, leading on to the restoration of the pharyngeal muscle activity and the airways open. So this cycle gets repeated. Now the various risk factors which are there are obesity with the body mass index being more than 30, increasing age, gender male to female 2:1, whereas in India we have recorded it to be 3:1. Nasal obstruction, neck size being more than 17 inches in males and 15 inches in females, craniofacial abnormalities, family history, alcohol or sedative use, smoking, allergies and associated conditions. So this is a plot between the frequency of decrease in the saturation. More than 4% arterial desaturation dips which is occurring in relation with the normal neck circumference, percentage predicted. So there is a point where there is further increase in the neck circumference leads on to more dip in the saturation, the frequency increases. The age, this is the relation showing with age, the risk factor being age, that is blue is the females and yellow is the males. So as the age advances, the percentage of individuals with AHI which is more also increases. It is a condition more common in males being shown 2:1 for males, but in India, as I said, there is 3:1 which has been shown. Then with the nasal obstruction, there is a predisposition, anatomical abnormality, even if it is not there, some obstruction as nasal congestion can also lead to temporary snoring and airway collapse. But with the nasal obstruction, what will happen is that the person will try to breathe from the mouth and increase the air resistance in the pharyngeal structures. As a result, to overcome this resistance, there is increase in the work of the breathing and airways intend to close more with obstruction then. Then the family history, that is those who have got more than 3 relatives as having obstructive sleep apnea have a more chance of developing the disease as compared to those with 2 or 1 relatives having obstructive sleep apnea. Then use of sedatives also is associated. Why? Because it affects the hypoglossal nerve output to the genioglossus muscle, and the muscle then is relaxing or is not able to maintain the tone and the patency. This is the record which is shown at this point. Diazepam injection was given and the phrenic nerve record and the hypoglossal nerve recordings were then taken for the muscle. The use of the alcohol also brings about similar changes in the hypoglossal output going to the genioglossus muscle. As the levels increase, it brings about more reduction in the output. Then as a result of smoking, again the chances of having obstructive sleep apnea are more. This is more for the current smokers as compared to former smokers. In addition, these are the associated conditions: hypothyroidism, acromegaly, amyloidosis, vocal cord paralysis, Marfan syndrome, neuromuscular disorders, hypertension, metabolic syndrome and diabetes which could be associated. When there is sleep apnea, there is intermittent hypoxia and the sleep fragmentation because the person is getting up repeatedly, there are arousals. As a result of that, there is excessive daytime sleepiness and tendency to have cardiovascular complications, because during the periods of apnea, there is increase in the oxidative stress and also sympathetic activation which occurs. All of these lead on to increase in the morbidity and mortality. In addition, there is excessive daytime sleepiness, which could again be of pathophysiological significance because it would lead to increased motor vehicle crashes, work-related accidents, poor job performance, depression, family discord, and decrease in the quality of life. Even the number of automobile accidents increase because the person generally tends to fall asleep even while on the wheel. So as a result, these individuals have more chances of having accidents as compared to those who are not having any apnea, or in general if we take all the drivers in a single group. And these road traffic accidents tendency will increase further if these patients are taking alcohol. So the chances of having the accidents increases further. These obstructive sleep apnea could lead on to cardiovascular morbidity, and the various conditions which have been further associated are systemic hypertension, cardiac arrhythmias, myocardial ischemia, cerebrovascular diseases, and pulmonary hypertension. This was just a brief pathophysiology in general of OSA. I'm sure in today's later and for tomorrow also, you will be having more lectures and the inputs regarding what happens in OSA with stroke, with myocardial problems or other conditions. So this is just showing that when these three things happen, intermittent hypoxia, then there is the pressure changes and the fragmentation of sleep, there is oxidative stress and sympathetic stimulation, and in addition, this directly, the pressure changes also will bring about endothelial dysfunction. So all of them together bring about increased cardiovascular mobility. So this is in brief, all predispositions occur for these conditions. There is increase in the leptin with resistance, increased insulin resistance, increased atherogenesis and relation with cardiovascular condition and with stroke, and increased atrial natriuretic factor, which will be related to nocturia in these patients. So thank you. I've tried to cover, maybe many aspects are still left uncovered.
Thank you, ma'am. That's the last session before lunch. So lunch is being arranged at Sikapiachi Auditorium basement, which is right opposite the college block. And will please be back in half an hour.